Assalamu alaikum dear students, welcome to the course Principle in Animal Life 1. This is lecture number 46. The topic is cellular respiration and fermentation. This will be the second part of the topic. Discussing agenda for this portion will be stepwise energy harvest via NAD and the electron transport chain, NAD molecule, NADH molecule, lecture outcome. After watching and listening this lecture, students will know about cellular respiration and fermentation and related concepts. Introduction. Stepwise energy harvest via NAD and the electron transport chain. So this whole chapter is about cellular respiration, and we will discuss each stage of aerobic cellular respiration in details. Here is a summary of how this process takes place and how this process leads to the formation of ATP. NADA is an oxidized form, and when it accepts electrons from the substrates during glycolysis or during Krebs cycle, then it becomes reduced and converted into NADH form. If energy is released from a cell all at once, it cannot be harnessed efficiently for constructive work. Now the question arises here that why there are so many stages of deriving energy from glucose molecule or other organic molecules. Glucose molecules enter glycolysis. There are 10 enzyme catalyzed reactions, then pyruvate molecule is formed. At the end, pyruvate molecules enter into link reaction where they are converted into acetyl coenzyme A, and then acetyl coenzyme A enters into Krebs cycle where there are further 8 reactions, and then the NADH and FADH2 molecules produced, they deliver their electrons to a series of electron carriers in electron transport chain, and in this process ATPs are synthesized. So what is the need for so many reactions? Why not this glucose molecule delivers all its stored energy in just a single chemical reaction? So the reason is that if all the energy is released from a fuel all at once, then a major portion of this energy will be wasted as heat, and therefore it will not perform any work. Now this process is divided into so many steps, and the reason is that energy is produced slowly and gradually at a rate which is feasible for the cell to accumulate this energy slowly and gradually. For example, if a gasoline tank explodes, it cannot drive a car very far. So means if all the energy reserves stored in a molecule, all the energy is liberated at once, then a very small amount of that energy will be able to perform work, and rest of the energy will be wasted as heat. Cellular respiration does not oxidize glucose molecule in a single explosive step either. Rather, glucose is broken down in a series of steps, each one catalyzed by an enzyme. A key step: electrons are stripped from the glucose, as is often the case in oxidation reactions. Each electron travels with a proton, thus as a hydrogen atom. The hydrogen atoms are not transferred directly to oxygen, but instead are usually passed first to an electron carrier, a coenzyme called nicotinamide adenine dinucleotide, which we refer to as NAD. So NAD is a derivative of the vitamin niacin. This coenzyme is well suited as an electron carrier because it can cycle easily between its oxidized form, which is NAD, and its reduced form, which is NADH. Recall from the first part of this lecture that a molecule, when loses its electrons, becomes oxidized, and when it gains electrons, then this molecule becomes reduced. So NADH is the reduced form of NAD, and NADH is carrying high energy electrons, which can run the affairs in electron transport chain. So just like the name suggests, electron transport chain, in this chain, which takes place in eukaryotes in the mitochondrial matrix, there are a series of electron carriers, and they are arranged in a linear sequence. The electrons flow from one carrier to another. So from where these electrons come, these electrons are provided by NADH and FADH2, and from where NADH and FADH2 picks those electrons, these electrons are delivered to NADH, NAD and FAD by the substrates of these reactions in glycolysis and Krebs cycle. As an electron acceptor, NAD functions as an oxidizing agent during respiration. Recall from the previous video the oxidizing agent and the reducing agent. Oxidizing agent is one which accepts electrons and thereby oxidizes another molecule. Now, how does NAD trap electrons from glucose and the other organic molecules in food? Okay, so we know that NAD is in its reduced form converts to NADH, and NADH carries high energy electrons. So how these electrons are transferred actually from the organic food or organic fuel to the NAD? Now this is NAD as an electron shuttle. So how the NAD molecules deliver electrons to the electron transport chain. Now the full name of NAD is nicotinamide adenine dinucleotide, and the figure describes its structure. Also the name describes its structure, which is the molecule consists of two nucleotides. This dinucleotide refers to two nucleotides, 1 and 2. And these two nucleotides are joined together at their phosphate groups. So nicotinamide is a nitrogenous base, although not one that is present in DNA or RNA. So nicotinamide itself is a nitrogenous base, but this nitrogenous base is not part of the nucleic acid. The enzymatic transfer of 2 electrons and a proton from an organic molecule in food to NAD reduces the NAD to NADH. Most of the electrons removed from food are transferred initially to NAD, forming NADH. So in the glycolysis, 2 NADH molecules are produced, then 1 NADH molecule is produced when the pyruvate converts to acetyl coenzyme A, and then in a Krebs cycle 3 more NAD molecules and 1 FADH2 molecule is produced for 1 pyruvate. So in the glycolysis, link reaction and Krebs cycle, a total of 10 NADH molecules and 2 FADH2 molecules are produced, and then these high energy electron carriers, they carry those electrons to the electron transport chain. So now this is the structure of nicotinamide adenine dinucleotide. It consists of 2 nucleotides and 2 sugar molecules, and to the sugar molecules the phosphate is attached. So they accept 2 electrons and thereby they become reduced. The enzyme which catalyzes this reaction is dehydrogenase, so which transfers the hydrogen group to NADH. Enzyme called dehydrogenases remove a pair of hydrogen atoms, usually 2 electrons and 2 protons, because a pair of hydrogen atoms has 2 electrons and 2 protons. So the dehydrogenases transfers these 2 electrons and protons from substrate. Substrate is one of the intermediates in a catabolic reaction, which is quite often glucose or its intermediate stages, and when the substrate loses its electrons, it becomes oxidized and NADH is reduced. Now the enzyme delivers the 2 electrons along with 1 proton to its enzyme coenzyme, which is NAD, forming NADH. The other proton is released as a hydrogen ion into the surrounding solution. So now recall this process again. 2 hydrogen atoms combine with NAD. The 2 hydrogen atoms passes their 2 electrons to NAD plus 1 proton to NAD, and one of the proton is released, and the NAD now has 2 electrons of the 2 hydrogen atoms and 1 proton, so it becomes NADH loaded with 2 electrons. By receiving 2 negatively charged electrons but only 1 positively charged proton, the NAD has the nicotinamide portion of NAD has its charge neutralized when NAD is reduced to NADH. So because it is lacking 1 electron, so when 1 electron joins it, its positive charge is removed, and then 1 more electron and 1 more proton also neutralizes the charge, and overall NADH is in this form without any charge superscript. The name NADH shows the hydrogen that has been received in the reaction. NAD is the most versatile electron acceptor in cellular respiration and functions in several of the redox steps, reduction oxidation steps, during the breakdown of glucose. So first, NAD molecules are reduced during glycolysis, link reaction, and Krebs cycle, and then they deliver their high energy electrons to electron transport chain. Thereby they are oxidized, and once again they are in a position to accept electrons, so they once again rush back to participate in glycolysis and Krebs cycle as electron acceptors. Electrons lose very little of their potential energy when they are transferred from glucose to NAD. Each NADH molecule formed during respiration represents stored energy that can be tapped to make ATP when the electrons complete their fall down an energy gradient from NADH to oxygen. If you refer to electron transport chain, which takes place in the mitochondrial matrix, a series of electron carriers are arranged in a sequence, just like 10 or 15 people are standing in a row. One person fills a bucket from a stream, and that person delivers this bucket to another person, and then this bucket all the way flows to a fire, and it is put up on that fire. So in this process, the NADH delivers its electrons, and then these electrons are transferred from one carrier to another carrier to yet another carrier, and then finally these electrons are joined with oxygen, and oxygen picks up one proton from the inner matrix and becomes water. So during this process, the energy released in these redox reactions is used to draw protons into the intermembrane space, and as the protons from the intermembrane space moves again into the lumen through ATP synthase. So in this process, ATP synthase catalyzes the synthesis of ATP from ADP. Now, by receiving two negatively charged electrons but only one charged proton, how do electrons that are extracted from glucose and stored as potential energy in NADH finally reach oxygen? It will help to compare the redox chemistry of cellular respiration to a much simpler reaction, the reaction between hydrogen and oxygen to form water. So in the very last step of aerobic cellular respiration, the electrons are delivered to oxygen atom, which is of course highly electronegative. So the oxygen atom, after picking electrons, also picks up one proton and becomes water molecule. Now mix hydrogen and oxygen in their molecular form and provide a spark of activation energy, and the gases combine explosively. In fact, combustion of liquid hydrogen and oxygen is harnessed to help power the rocket engines that boost satellites into orbit and launch spacecrafts. So this is the hydrogen molecule, and it combines with oxygen. Of course, oxygen cannot exist freely in the atmosphere. It must bind with another oxygen. Therefore, you have this value which represents that it is a single oxygen, because two hydrogen atoms combined with one oxygen atom to form water. Now this reaction requires some kind of input of energy, and explosive release of heat and light energy takes place when they combine with each other. So remember that this is an exergonic reaction, and the free energy of the reactants is greater than the products. In exergonic reactions, the free energy of the reactants is always greater than the products. Now, so in the previous slide, we were referring to a process which is uncontrolled. So the same process also occurs in aerobic cellular respiration, but it is highly controlled and coordinated. So the explosion represents a release of energy as the electrons of hydrogen fall closer to the electronegative oxygen atoms. Cellular respiration also brings hydrogen and oxygen together to form water. In the very last step of aerobic cellular respiration, this process occurs in electron transport chain. But there are two important differences. The first difference is in cellular respiration, the hydrogen that reacts with water is derived from organic molecules rather than H2. Second, instead of occurring in one explosive reaction, respiration uses an electron transport chain to break the fall of electrons to oxygen into several enzyme releasing steps. So remember from where the NAD picked their electrons. NAD picked their electrons from the hydrogen atoms which were present in the organic food molecules, and it is these electrons which will be delivered to electron transport chain, and those electrons will flow from one carrier to another carrier to yet another carrier, and finally they will become attached with oxygen forming water. So simply the process is same, the combination of hydrogen with oxygen to form water. But in case of cellular respiration, this process does not take place all of a sudden. This process takes place in a series of several steps. So you can see here the food molecules or the substrate deliver their electrons to NAD, and then NAD passes those electrons to a series of electron carriers, and finally these electrons are combined with oxygen, and oxygen also picks up protons from the matrix and form water. An electron transport chain consists of a number of molecules, mostly proteins, but in the inner membrane of the mitochondria of eukaryotic cells and the plasma membrane of respiring prokaryotes. So electron transport chain, it consists of a series of proteins, and these proteins are embedded in the inner membrane of mitochondria in eukaryotes. And because prokaryotes lack mitochondria, so if the prokaryotes are aerobic, then they are also supplied with those proteins embedded in the plasma membrane. So in prokaryotes, the process of aerobic cellular respiration also takes place, that is aerobic prokaryotes. Electrons removed from glucose are shuttled by NADH. They are carried by NADH to the top high energy end of the chain. At the bottom lower energy end, oxygen captures these electrons along with hydrogen nuclei, forming water. So electron transfer from NADH to oxygen is an exergonic reaction with a free energy of 53 kilocalories or 2 to 2 kilojoules per mole. So during this joining of electrons with oxygen is an exergonic reaction where that much energy is produced, and remember, free energy is the energy available for performing work. So this energy is available to the cell to perform work. Now, instead of this energy being released and wasted in a single explosive step, just like in the previous slides, here in the uncontrolled reaction, the hydrogen and oxygen combined with one another instantaneously, all of a sudden, and it results in an explosion where heat and light is released. So in this process a lot of energy is wasted which cannot be utilized to perform work. On the other hand, in cellular respiration the same process takes place, but it takes place in several steps, and energy is liberated slowly and gradually. As the electrons are passed from one carrier to another carrier, some energy is released in this process, and that energy is used to perform work to synthesize ATPs. Now, instead of this energy being released and wasted in a single explosive step, electron cascade down the chain from one carrier molecule to the next in a series of redox reactions, losing a small amount of energy with each step until they finally reach oxygen, the terminal electron acceptor, which has a very great affinity for electrons. So what is affinity? Affinity refers to the ability of an atom to attract a pair of electrons towards itself. So this oxygen, because of possessing high electronegativity, has higher desire for electrons, and it is the final acceptor of electrons. Now each downhill carrier has a greater affinity for electrons than, and it is thus capable of accepting electrons from oxidizing its uphill neighbor with oxygen at the bottom of the chain. So the electrons in electron transport chain are transferred from one carrier to another carrier to another carrier. Now suppose one of the electron acceptors which is present here, it has less electronegativity. This one has more electronegativity than this one, and this one has more electronegativity than this one. So the electronegativity increases as the electrons pass from one carrier to another carrier. The successive carriers have more electronegativity, and this whole story ends with oxygen which has the highest electronegativity values of all the carriers in the chain. So as the electrons move from one carrier to another carrier, these electrons loses part of the energy. Now, therefore, the electrons transferred from glucose to NAD, reducing it to NADH, fall down an energy gradient in the electron transport chain to a far more stable location in an electronegative oxygen atom from O2. Now put another way, oxygen pulls electrons down the chain in an energy-yielding tumble analogous to gravity pulling objects downhill. In summary, during cellular respiration, most electrons travel the following downhill route. So first, glucose is broken down, molecules of NADH are produced, pyruvate is broken down, in Krebs cycle molecules of NADH are produced, and then these NADH molecules, they shuttle their electrons to electron transport chain, and then electron transport chain has oxygen at its end, so these electrons becomes part of oxygen forming water, and in this process energy is liberated, which is used to draw electrons into the intermembrane, draw protons into the intermembrane space, and then these protons return back to the matrix via ATP synthesis and ATP is generated. So later in this chapter you will learn more about how the cells uses the energy released from this exergonic electron fall to regenerate its supply of ATP. In electron transport chain you will study this process in detail. For now, having covered the basic redox mechanisms of cellular respiration, let's look at the entire process by which energy is harvested from organic fuels. So this process of aerobic cellular respiration comprises of three major steps, and these three major steps are glycolysis, in which a glucose molecule, a 6-carbon molecule, is broken down into 3-carbon molecules called pyruvic acid, and glycolysis takes place in cytoplasm. This process does not require oxygen, and it is a universal energy deriving pathway. It is thought to be the most ancient energy deriving pathway, and it produces a net of 2 ATPs and 2 NADH molecules. So the end product of glycolysis, which is pyruvate is converted into acetyl coenzyme A through link reaction. It is a process which is catalyzed by the enzyme pyruvate dehydrogenase, and there are a total of 3 steps which converts pyruvate to acetyl coenzyme A, and then acetyl coenzyme A enters citric acid cycle. In citric acid cycle, 3 more NADH and 1 FADH2 molecule is produced for each molecule of pyruvate, and then all these NADH and FADH2 produced, that is 10 NADH and 2 FADH2 produced in the first 2 steps, they shuttle their electrons to electron transport chain, where the major bulk of ATPs are produced through oxidative phosphorylation. In the first 2 stages, 4 ATPs are also produced, but they are produced through substrate level phosphorylation. Biochemists usually reserve the term cellular respiration for stage 2 and 3 because these stages require oxygen, so therefore they usually refer this term to the second and third stage. In this text, however, we include glycolysis as a part of cellular respiration because most respiring cells deriving energy from glucose use glycolysis to produce the starting material for the citric acid cycle. So although the last two stages are oxygen requiring, but the last two stages are only possible if glycolysis provides the raw material. So glycolysis, although this process occurs independent of oxygen but is categorized in cellular respiration for the fact that it provides raw material for the later two stages. As programmed in figure 7, glycolysis and then pyruvate oxidation and the citric acid cycle are the catabolic pathways that break down glucose and other organic molecules. Now glycolysis, which occurs in the cytosol, begins the degradation process by breaking glucose into two molecules of compound called pyruvate. So this is the process. Glycolysis breaks down glucose into two molecules of pyruvic acid or pyruvate. Then this pyruvate enters mitochondria where pyruvate oxidation takes place, where the process of oxidative decarboxylation converts pyruvate to acetyl coenzyme A. Then this enters into Krebs cycle or citric acid cycle, which produces more NADH and FADH2, and then all these electron carriers shuttle their electrons to electron transport chain. 2 ATPs produce here, 2 ATPs produce here, and from 26 to 28 ATPs produced here. ATPs produced here are through oxidative phosphorylation. ATPs produced here and here are through substrate level phosphorylation. So during glycolysis, each glucose molecule is broken down into two molecules of pyruvate in eukaryotic cells. As shown here, the pyruvate enters the mitochondria. There it is oxidized to acetyl coenzyme A, which will be further oxidized to carbon dioxide. In the citric acid cycle, the electron carrier NADH and FADH2 transfer electrons derived from glucose to electron transport chain. During oxidative phosphorylation, which takes place in the inner lumen of mitochondria, electron transport chain converts the chemical energy to form used for ATP synthesis in the process called chemiosmosis. Chemiosmosis is the process in which the protons present in the intermembrane space, they try to enter back into the matrix through ATP synthase enzyme. So this process is referred to as chemiosmosis. Now in the third stage of respiration, which is electron transport chain accepts electrons from the NADH and FADH2 that were produced previously in the first two stages, and then these two high energy electron carriers deliver their electrons to a series of electron acceptors, and these electrons move down the chain from one acceptor to another acceptor, and there are a series of redox reactions where one acceptor is first oxidized and one acceptor is first reduced, and then when it delivers its electrons to another carrier, then it is oxidized and it reduces the another carrier, and this process occurs once more up till the stage when the electrons are finally accepted by oxygen forming water. So at the end of the chain, the electrons are combined with molecular oxygen and hydrogen ions forming water. The energy released at each step of the chain is stored in a form the mitochondria can use to make ATP from ADP. So this mode of ATP synthesis is called oxidative phosphorylation because it is powered by the redox reactions of the electron transport chain. The redox reactions taking place in the inner lumen provides energy for pumping the protons into the intermembrane space, and then these protons return via ATP synthase and ATP is synthesized. Now in eukaryotes, pyruvate enters the mitochondria and is oxidized to a compound called acetyl coenzyme A, which enters the citric acid cycle. There the breakdown of glucose to carbon dioxide is completed. Remember that pyruvate is a 3-carbon molecule and the end product of glycolysis is 2 pyruvate molecules. So even after glycolysis, all the 6 carbon molecules which are present in glucose are still intact in 2 pyruvate molecules. Then when the pyruvate converts to acetyl coenzyme A, 1 carbon is liberated as carbon dioxide, and then in Krebs cycle 2 more carbon molecules are released as carbon dioxide. So in Krebs cycle, all the carbon molecules which are present in pyruvate are liberated as carbon dioxide. So there is a complete breakdown of glucose which is completed in Krebs cycle. So thus the carbon dioxide produced by respiration represents fragments of oxidized organic molecules. Now the carbon dioxide which is exhaled to the environment is actually a result of this breakdown of glucose. Some of the steps of glycolysis in the citric acid cycle are redox reactions in which dehydrogenases transfer electrons from substrate to NAD or the related electron carrier FAD, forming NADH and FADH2. You will learn NADH about NADH and FADH2 during our lectures on glycolysis, Krebs cycle and electron transport chain. In the third stage of respiration, the electron transport chain accepts electrons from NADH or FADH2 generated during the first 2 stages and passes these electrons down the chain. So in eukaryotic cells, the inner membrane of the mitochondria is the site of electron transport chain and another process called chemiosmosis, together making up oxidative phosphorylation. Oxidative phosphorylation accounts for 90% of the ATP generated by respiration. 2 ATPs are produced during glycolysis, 2 ATPs are produced during Krebs cycle, and a major chunk of ATPs are produced in mitochondria through oxidative phosphorylation. So if you represent those percentages in figures, then it makes up to 26 to 28 ATPs produced during electron transport chain in mitochondria. So a small amount of ATP is formed directly in a few reactions of glycolysis, that is 2 ATPs, and then 2 ATPs in citric acid cycle, but these 4 ATPs are produced through substrate level phosphorylation. When a substrate, an intermediate of a metabolic pathway, provide phosphate group to ADP to form ATP. Now this mode of ATP synthesis occurs when an enzyme transfers a phosphate group from a substrate molecule to ADP rather than adding an inorganic phosphate to ADP as in oxidative phosphorylation. So substrate molecule here refers to an organic molecule generated as an intermediate during the catabolism of glucose. You will see examples of substrate level phosphorylation later in this chapter during our lectures on glycolysis and electron transport chain. You can think of the whole process this way. When you withdraw a relatively large sum of money from an ATM, it is not delivered to you in a single bill of a large denomination. Instead, the machine dispenses a number of smaller denomination bills that you can spend more easily. This is analogous to ATP production during cellular respiration. So now they have compared this process to the ATPs produced through aerobic cellular respiration, just like when you want to draw money from ATM. Suppose in our country only 50,000 can be drawn from ATM, but in other countries you can draw as much as you like, but if you put a number into ATM device, all the money is not delivered to you in a single episode, but in 2 or 3 episodes, which you can collect and count easily with peace. So similar is the case with the production of ATPs during aerobic cellular respiration, where the energy is generated slowly and gradually at a pace which can be accumulated easily by the cell. For each molecule of glucose degraded to carbon dioxide and water by respiration, the cell makes up to about 32 molecules of ATP, each with 7.3 kilocalories per mole of free energy. So each molecule of ATP carries that much energy. So if you want to calculate the amount of energy produced in kilocalories per mole from a single glucose molecule, then you multiply this with 32. Respiration caches in the large denomination of energy banked in a single molecule of glucose, 686 kilocalories per mole under standard conditions for the small change of many molecules of ATP, which is more practical for the cell to spend on its work. So just like you have a currency note of 5,000 and then you go to a bank and the bank gives you several notes of 500 rupees or 100 rupees, and you can use that smaller notes much more conveniently. So this process is also relevant, where the energy is produced and stored in molecules called ATP, and then ATP can be used as per the need of the cell. This preview has introduced you to how glycolysis, the citric acid cycle, and oxidative phosphorylation fit into the process of cellular respiration, so you can keep the big picture in mind as you take a closer look at each of these 3 stages of respiration. So here you studied a sort of summary of this whole process, and then of course there will be lectures delivered for each stage separately. So with that, we come to the end of this lecture. Remember, this lecture was divided into two parts, so in order to know about the complete lecture in detail, kindly go through both these videos. Thank you and Allah Hafiz.